Hello everyone, my name is Dr. Christine and I work here at the BioArte under the leadership of Dr. Manuele Biazzo. Today we're going to speak about the skin. So when we look at the skin, we just see the surface, but underneath at the microbial level, it is highly complex ecosystem of bacteria, fungi, viruses and phages. They all interact within our immune system. This community is not just random, it reflects health or disease. Until now, our ability to truly study it has been very limited. Most sequencing technologies capture only a fragment. They identify the species, but stop on the strain level resolution. And that's where the clinical difference lies. For example, two strains of Cutibacterium acnes, for example, can have opposite roles. One can be protective and the other one can be highly pro-inflammatory. Without strain level insight, we are essentially looking at the skin health with a blurred vision. This is exactly why we developed at BioArte the Skin BioArte Microbiota. It is a platform that combines three pillars: long-read sequencing, which gives us a complete genome from skin swabs, a proprietary base calling pipeline optimized to handle the noisy data of skin samples, and three, a custom bioinformatics algorithm that assembles, classifies, and interprets microbial communities in unprecedented detail. With the Skin BioArte Microbiota, we can detect and distinguish specific strains such as Cutibacterium acnes linked to acne versus those that are commensal. Staphylococcus aureus, for example, which colonizes in eczema. Shifts in diversity that predict inflammation or treatment response. And even functional genes such as antibiotic resistance, virus factors, or metabolic traits that explain why certain strains behave the way they do. We first tested the system in a proof of principle study. The question was, can our platform discriminate healthy skin from dysbiotic skin? The answer was a clear yes. The microbial signatures were distinct and reproducible, providing the concept. Encouraged by this, we launched an observational study in patients with acne. Here we applied our own product Skin Duo to real clinical samples, tracking the skin microbiota of patients over time after treatment. For the first time, we could map acne, not just the presence of Cutibacterium acnes or not, but the strain level patterns and genetic factors that those strains carried. This allowed us to identify microbial fingerprints of acne severity and treatment response. The outcome was not just a diagnostic. Inside the Insight, Skin Duo actually guided intervention that ameliorated acne, detecting and targeting the problematic strains while preserving or even supporting the protective ones. We saw a measurable improvement in patient skin condition. We began as a sequencing and computational challenge that has become a therapeutic opportunity. We are no longer limited to describing the skin microbiota. We can detect and interpret and ultimately modulate it to improve the skin health.